Okay, welcome everybody. Thank you for coming. It's 5 o'clock. I know this course meets at an unusual time. I hope it's not too inconvenient for people. We normally will have a short break in the middle that I hope is helpful. In the days when we did it in person, we did a food break in the middle, but can't do that online. I guess you could bring something for yourself to keep you going. This course also is very much a course that responds to the students. And each time we give it, it's a slightly different group of people, a slightly different set of backgrounds and interests. And so I try to make the course as responsive as possible to that. Today, we're going to talk a little bit about class organization. Then I'll do a brief review about why we need this kind of modeling and what these kind of models are. We'll have some people present some possible ideas for projects that they could help with. You're not by any means responsible for doing it that way, but it's an opportunity. Then I'll give some examples of this kind of modeling. We'll do a little bit of running of models. We may or may not today to get to a model that Furkan wrote 2 years ago. That is a great demonstration of what this kind of modeling can do. And then we'll do some more examples and maybe do some more runs together. And then we will talk a little bit about projects. This semester, I want to emphasize even more than in the past that this is going to be a project-based course. I really would like people at the end of the semester to have a simulation that they could put up on this Nanohub public server that will attract people's attention and get you a publication and be something that you're proud of. People who took my course in the fall, all of you did a great job putting up Nanohub apps this year. It worked out really well. I learned something from the experiences of successes and failures in that, which I'll try to apply this semester, and you'll have to help keep me on the straight and narrow about that. We're going to meet, as usual for these classes, just before 5 till just before 8. The reason we have a single block of time and it's long is because a certain amount of the time we'll be spending doing computing. So in the sense, it's a mixture of a lecture and a lab course. And we need to have, it's much more efficient to do that in a single block. We don't have a formal AI for the class this semester, but we will have help, at least at the beginnings of the evenings, from TJ Seago, who's the lead developer for the software we're using, currently developer for the software, and also a very accomplished modeler. Joshua Aponte Serrano, who people know for last semester. He was the teaching assistant for last semester, will be helping us out some. Giuliano isn't formally going to be helping us, but he is an expert on getting things running on Nanohub, and also helps develop CompuCell and uses it. So he'll be available. Furkan, if he sticks it out with us, is an expert on this too, I should say. And we'll have access at least some of the time to Maciek Swat, who was the person who wrote CompuCell in the first place. He's not going to be on the calls, but he's very responsive to emails if there are issues. We're going to be basing a lot of the materials that we're going to do this semester on the summer course version of this course. We teach a week-long course on this kind of modeling every year. It starts at 9 in the morning and runs till 6 p.m. for 5 or 6 consecutive days. It's very intensive. We develop a lot of materials. I'm going to try to be doing it, not a fully flipped classroom, but I'm going to try to ask people starting next week to watch some of the videos that we recorded from the summer and do exercises from those so that we could use more of the class time to work on projects. That's going to be a little bit of an experiment. If it doesn't work, we'll go back to a more classic lecture format. But we have recordings of all of the not identical versions of the lectures, but versions of these lectures online, which you'll have access to, as well as specific homework problems and solutions. So to some extent, this could be a self-taught course if you wanted to, although it's could certainly be improved. The requirements for the course, if people want to know about that, are going to be some homeworks. There may be slightly fewer this semester than in previous versions of this course. The main project will be a project simulation, which will be a documentation for it, which will be released on Nanohub as a publicly available, usable software package. That's going to be the main project. You'll do an oral presentation of that. I'm going to ask people also to do a video introduction to their project and upload it onto YouTube and Nanohub as a way of advertising the work you've done. Because if you do all that work, I mean, this is basically a result of people spending lots of time building very beautiful models, and then those models are turned into a term paper, they're handed in to me, and nobody sees them. And if we do them this way, once your model is posted on Nanohub, it gets a DOI, it counts as a publication. You can show it to your friends, your family, your potential employers, and so on. And so it's a little more work to do it that way, but it also means that you're advancing your career in a way that writing a term paper doesn't. And so it worked out quite well in the fall using a slightly simpler software package. I hope it'll work today well here. I know some of you have done this in other classes, especially ones you've done with Vikram, perhaps. If you've done a course with Vikram or with Paul Macklin, if you've worked with him. And so you can also help us on our end thinking about the best ways to put this forward. Something that we're going to try to do this semester, and it's going to be tricky. This semester is only 14 weeks. The university has cut the length of the semester back by 2 weeks. That's going to be tough. I'm going to try to ask people to get preliminary versions of their projects done a couple of weeks before the end of the semester so that we can do rounds of revision and critiquing. Last semester, we found that it was really helpful to get your feedback from classmates to work on projects and improve them. But we ran out of time because of the end of the semester. So I'm going to ask people to really push and try to get something done early enough that you can present it. We can do formal critiques as in a project course. You can do a revision, and that way when you put this up for the public, it's been worked through by your classmates. I think that's a really good discipline to have. There won't be any exams. And some people I know in advance had said, well, some people said they have more expertise in biology or mathematics than programming. The projects can be done in teams. And so it's perfectly okay if you think that you are strong, for example, in biological background, find somebody who is more of a software person who's not so strong on the biology side and work with them if you can find a project. You could have 2 people or 3 people on a project, or team with one of the senior people that is going to be on this call pitching ideas. Okay. So the tentative schedule is this introductory lecture here. We're going to start talking about possible projects already today. I'd like to start meeting with people as soon as possible in small groups or individually to talk about possible projects. Next week, we'll introduce some of the basic concepts in CompuCell, things like how you control cells, volume, adhesion, motility, and so on. The next week, we'll do cell division, which probably most of the projects need. Diffusion the week after that, and then we'll come up with more specialized topics. And I'd like to have basically a check-in where every group every other week presents their progress, because if we get behind on projects, it's going to be quite difficult to catch up. For some reason, the university has random holidays this semester, so the 16th of February, we don't get to meet. You get the day off. I hope it's valuable to you. And so I was hoping that we would actually be able to get the initial project designs done in the middle of February, and then actual first rounds of the projects around the last week of March. And that would give us 4 weeks to do revisions and final uploads. Depending on how many teams there are, we may need 2 weeks to go through the projects, in which case this is written that way. If people team up in larger teams and we can get through everything in 1 week, then we'll have a little bit more flexibility on schedule. In principle, the 4th of May is exam week. If we need that to do final presentations, we'll hold that time. But if we get through in time, we won't. But if you count, we count 14 weeks of class, which is short. So I hope people understand that. Are there any questions about that class structure so far? People who've had a class with me before will recognize this as a sort of an evolution of the approach that I've taken before. Some people have, some people haven't. I want to emphasize that there's no requirement that you've taken the other class, the fall version of this class. It's somewhat helpful to have taken that class because you'll be familiar with some of the material, but there's no reason you don't need it. The few things that you need from last semester, we will cover in this semester. Okay, so there is no formal textbook. That's a plus and a minus. There are online manuals for the software. TJ is our manual writer, along with Maciek. He would love it if you look at the manuals and make critiques and suggest improvements to them. There's a quick reference guide that will help you get started with the software. The main manual will be the Python scripting manual because Python is the language we use to write simulations for the most part. There's a generic reference manual. These are all available at www.CompuCell3D.org. If you want more on the modeling methodology, there's a textbook by Shana and Preziosi called Cellular Models, Multiscale Extensions and Biological Applications. That's from 2013. It's a little bit old. And there's some other textbooks here on general topics in mathematical and computational biology. Depending on your background, they may or may not be of interest to you. Herbert Sauer's Systems Biology is a textbook we've used in the fall semester course. I think it's pretty good. Nicholas Britton, Essential Mathematical Biology is good background for mathematical biology. We won't do a lot of that in this semester. There's the classic Scott Gilbert and Singer developmental biology textbook. If you want to brush up on your basic biology, it's a very good thing. You could buy a slightly out-of-date edition for $5. You don't need the latest edition for $200. Development hasn't changed that much. A lot more molecular species names in the newest versions. And trying to think if there's anything else that we really need to go over about this here. We'll have, if you go to the CompuCell website, there are a couple of reference papers. There's a particular one on CompuCell and modeling with CompuCell that's pretty useful. But your best resources will be your classmates, me, and the other people who will be on this call today. The programming side of things is not people who took the fall course. There's a little bit more programming involved this time than there was last semester, but it's not very demanding in terms of Python, although Furkan can tell us whether he thinks that's true or not. Occasionally, there'll be Python annoyances that we'll come back to, and we'll do our best. And the course topic is essentially life, in particular multicellular life, and how cells organize to form tissues and how they work. And that really is the point of the course. And we could think about why we need to do modeling at all. And we're going to think about it again in the context of how you build an organism, which is my own area of background and interest. How an organism maintains itself. It's called homeostasis, which is something that people often don't think about so much. That every cell in your body pretty much gets replaced during your lifetime, except for your neurons and a few others. Even the non-cellular tissue, the material in your bones is replaced on a time scale of about 15 years. So actually your body is highly dynamic, even if you don't seem to be changing. There's a continual flow of material and energy in and out of your cells. There's continual death of cells, birth of cells, reorganization of cells. And something that we've become very aware of in the past year is that there also are continual challenges to your body, in particular things like infectious diseases. And so last spring, traditionally this course has been taught around modeling cancer. Last spring in March, the student said after spring break, can we make the course about COVID instead? And so a couple of the students, undergraduates in the student course, one of the graduate students, one master's student, a couple of undergraduates, worked with us over a couple of days, wrote a COVID model, and that is something that we continue to work with, and that'll be something people could work on if you're interested. We've also worked in the area of toxicology. It doesn't sound so sexy when you swallow a chemical and damages your tissue. How does it do it? But it's an area of importance, and it's something we've worked with the Environmental Protection Agency, US EPA, quite a bit on. The EPA uses our software in that context. And then potentially, and TJ is particularly interested in this, is bioengineering. How do you design tissues? It's easy to do tissue printing these days. Getting the printed tissues to do anything useful is hard. Basically, you could put cells in place, but what they do afterwards is what they do. And potentially one would like to be able to model that. In fact, the very first version of CompuCell was written as part of a grant back when I was still on the faculty at Notre Dame. One of the people on the grant was somebody named Gabor Forgax. And he founded the company called Organovo, which is the first tissue printing company. That actually grew out of the CompuCell project. So ironically, we didn't ever do very much modeling of his work. We started out that way, but never went back to it. So there are a lot of questions we could ask for. I'm particularly interested these days in thinking about how you could use this kind of modeling to improve medicine. That's not certainly a requirement for your interests, but if you think about medical treatment today, it's rather medieval. In particular, it responds to problems after they occur. And in engineering, one of the goals of engineering is to prevent failures, or at least to prevent catastrophic failures. Instead of waiting for things to fail and then fixing them, we try to keep them working. And so one can think about the possibility of building computational models of individuals that would be able to predict problems and help you regulate states of health rather than responding to illness. There are also some technical issues that medicine fundamentally isn't scientific because you can't repeat experiments and you can't do control experiments. Each human is different, and what you want to know when you take a pill is what it'll do to you. The only way you can find out is by taking the pill. That means you can't do an experiment, or at least the only experiment you do is an uncontrolled experiment. You can't compare what would have happened if you took pill number 1 versus pill number 2, because you only take one. And you can't compare what would have happened if you took pill number 1 versus doing nothing. And so computational models in principle allow you to build controls and to be able to compare courses of action, which are very hard to do otherwise. as we're starting to find out in the work that Paul is doing on in-host viral replication and immune response and the work we're doing, we really don't understand the human immune system. We really don't understand the human immune system. And that means that when we try to do things like do immunosuppression for COVID patients, it's a shot in the dark. When they design COVID vaccines, you want to know, is the COVID vaccine going to give, how long does immunity last when you get vaccinated for COVID? The answer is we have no clue, really no clue. Why is it that some vaccines give lifelong immunity and other vaccines give you 3 months or 4 months of immunity? We don't know. Eventually, we'd like to have models of the immune system that are detailed enough that you could actually predict that instead of having to wait and see. We're not going to be able to get there in this course, but as a dream, I think it's worth holding in mind. If you were going to ask me what's a really good career, I'd say understanding the immune system is a good career. You're not going to run out of problems, and you're not probably run out of money either. You might start a $1 billion company if you can solve that problem. So those are some of the reasons one might want to be able to build predictive models. Some people call those digital twins. We tend to call them virtual tissues, but we're really talking about the same thing. Simulations that take a state of a system and predict how it's going to change in time. If you're unfamiliar with this world, haven't taken this course or version of this course before, I should say that as far as personalized medicine goes, there are lots of approaches to personalized medicine. There are all of the omics bioinformatics approaches to personalized medicine. There's a whole world of what are called cell atlases, where you're doing essentially genomics or proteomics at the level of single cells. And that's pretty amazing, but it's in fixed tissues. It's not in you. So you kill a human or a mouse, you slice them up, and you look at the genome or the proteome or something else in each cell. It gives you tremendous spatial resolution, but it's one point in time, and it's not you, it's somebody else. The question is, how do you take that information and use it to say something about you as a living individual? There are, of course, all sorts of wonderful deep learning and AI approaches that are essentially data-based. And I don't like the word data because data doesn't have really any meaning. You have to say what kind of data you're talking about. Mining of medical records, for example, is very interesting, can do a lot, but it doesn't give you a mechanistic understanding of how your body works. And so one of the things that I think is going to be very interesting going forward in this world is combining artificial intelligence with the kind of mechanistic modeling that we're going to talk about in this course. There are many ways you could do that. You could use it in parallel, where you have a mechanistic model and a data mining model. You could use artificial intelligence to do acceleration of computations that are quite expensive. We have Javier in my group, for example, has been doing AI-based diffusion solvers, which can speed up these calculations by a factor of 100 or more. Parameter identification and optimization, which are things that we probably won't get into too much in this course, but you'll run into if you continue in this field, are very challenging. And artificial intelligence methods can be very helpful in that context as well. And then on the side of experiment, we have people in the department like Feng who do organoid culture. So they build little tissues on a chip where you could control what's happening in more detail than you could in a living organism, whether that's a human or a mouse. But they're not exactly the same as the organs in vivo. And so how do you map the information you get from an organ on a chip to a real organ? Still not well understood. And then in the long term, there's been the promise of tissue engineering, which is building replacement parts for people. Still hasn't really lived up to the hope, but I think in the next 20 years it will. And if you're going to be able to engineer tissues, you're going to have to design them. And that means you're going to be able to predict how tissues behave. So those are some of the parallels. All of these are complementary approaches. We're going to need all of them to really deliver. So if we come back just to the very simple quantitative motivation, we could imagine some gene A, a gene A promotes, that increases the amount of gene B and gene C, and gene B increases the amount of D, but gene C decreases the amount of D. If we increase the amount of A, B and C fight each other over D, and we can't predict what happens to D. And so in that situation, we'd have to have some detailed quantitative understanding of what's going on to be able to predict what happens to D when we change A. In a tissue engineering context, cells crawl around. The way they crawl depends on the chemistry of what they're crawling on, the texture of what they're crawling on, and the stiffness of what they're crawling on. And you could ask the question, if I make the gel that the cell is crawling on stiffer, does the cell crawl faster or not? In general, we can't answer that. There are models that try to answer that. And then more tissue level organization questions. For example, when tumors grow, they cause blood vessels to grow into the tumor. How does that neovascularization, growth of new blood vessels, change the shape and evolution of the tumor? So those are the kinds of things that we want to be able to begin to think about in this class. And again, I'll point out that today, almost any complex engineered system has a digital twin. When you get in a jet plane, even if it's made by Boeing, there is a model of the engines, and those continually predict how that engine should behave. At the moment there's any deviation between that prediction and the measured behavior of that engine, a flag goes up saying something is going wrong, service the engine now. And the use of digital twins in jet engines has been one of the single biggest factors in increasing the safety of airplanes, because you catch problems in jets before the engine fails rather than when it fails. And you'd certainly rather fix it on the ground before it blows up while the plane is flying. And so that kind of digital twin predictivity is huge. And we use the word virtual. Sometimes digital twins are also called virtual twins. And the term virtual tissue was one that the EPA coined some years ago. It's not the universal way of describing this, but we like it as a concept. And ultimately, we dream of having digital replicas, first of components of your body and eventually perhaps of the whole body, that can predict how you're going to function, when you're going to get sick, what we can do to prevent the illness, and if you do get sick, how we can treat it effectively. This is all new. There are very few digital twin or virtual twin applications, although they're beginning to develop. If you go into the digital pancreas, has a very simple digital twin. It consists of something that measures your blood glucose. That was actually the hardest part of the digital pancreas. For a long time, there were implantable insulin pumps. People with diabetes would have a bump on them, which was an implantable insulin pump. But you had this funny thing where you had an implantable insulin pump, it was very advanced. The person would still be sticking themselves, getting a drop of blood, putting that drop of blood on a sensor, measuring their glucose level, typing it into a remote controller, and controlling the insulin pump. And the reason for that was that it was quite difficult to build implantable glucose sensors that would work for a long time and were accurate. Now there are, and so they're closed loop, what they call, again, artificial pancreas, where the implant measures the blood glucose level and then has a model of how your particular body responds to insulin, and then doses you with insulin in response. Pacemakers, cardiac pacemakers do that to some extent as well. The computational models are not that complicated, but they're an example of what happens when you combine implantable medical devices with modeling. And it would be really interesting to see how that develops in the future. As far as biological simulations go, we'll find that one of the problems in biology is that the range of length scales is enormous. There are some problems where the individual atoms or molecules matter in detail. And there are other ones, and then you have the scale of your whole body, which is meters. So you're talking about tens of nanometers to meters. And the time scale over which processes happen goes from milliseconds or microseconds for metabolic processes to weeks, years, or years for some kinds of developmental and disease processes. You can't model all of those levels in time and space in one model, but you do have to put together multiple modeling methodologies and multiple levels of model. In the world that we're going to occupy in this course, we're typically going to focus on 3 levels of modeling. We're going to have regulation, metabolism, and signaling at the subcellular level, represented not spatially, but using dynamic networks. That's what the course in the fall is about. But again, it's not a requirement for this course. You're going to have cells and their interactions, which are controlled by those networks. And then you're going to have how those cells organize themselves. I don't know why the movie isn't playing now. It was playing earlier, but not now. How those cells organize to form tissues. And sometimes we'll put that into a larger context where you look at transport in the whole body. Ironically, that's also a network model. So you have networks at the lowest and the highest scale. These are going to be deterministic models. So if you come from a bioinformatics background, it's going to look very different. They're additive models. That is, you start with nothing and you build up. That's the opposite of a data-based model. And we're going to be making movies, spatiotemporal movies. And so the outputs of our simulations will be movies of how tissues change. There'll also be time series, but the main thing you'll be getting out. So if we look at actual tissue development, this is a chicken embryo building the structures that are going to give rise to the spinal cord. This is an area we've worked on a lot in my lab over the years. This is a movie of every cell in a zebrafish embryo during early development. This is from a French group. We're currently working with a group at Chan Zuckerberg Institute in San Francisco that's doing this kind of imaging. Actually, the person who runs that group was a student of the person who did this work some time ago, trying to understand early embryonic development. This is work that was done by Maria Bondison before she came to Indiana University. These are blood vessels growing between the segments that you're seeing develop here on the right in a zebrafish also. And then this was some very beautiful work on branching morphogenesis and explanted lung tissue. So this is somebody trying to understand how the lung organizes itself during development. Now what you see is that the cells move around a lot, they change shapes a lot, they reorganize, the tissues fold, bend, rotate. So there's a lot of movement. In adult tissues, you don't have so much relative movement of cells, but in embryos, you have a lot of relative movement. And a lot of the modeling technologies that we've developed were designed to deal with that kind of movement and reorganization that occurs in tissues. It's not easy to do this. It's much, even a very simple living organism is much more complicated than the most complicated jet aircraft. In terms of the number of parts, in terms of the variety, in terms of the number of things you have to think about to control. All of these things are much more complicated in even simple living organisms than the most complicated human engineered systems. One of the biggest problems in biology is that things are not repeatable. In biology, you'll run into things like cell types, where you say these 2 cells are both neurons. No 2 neurons are the same. If I have 2 number 18 screws that are made out of brass, they're pretty much the same. And so when I build engineered systems, I have modular components that are interchangeable. When I move to biology, that modularity and interchangeability are not so good. There's some modularity, but the boundaries between modules are fuzzy, and the homogeneity of modules, one module to the next, isn't perfect. And that makes it a lot harder to do modeling. We certainly don't have the ability to model things in complete detail. There's no ab initio calculation. We're not doing physics. And that means everything we do is approximate, and it means we always have to challenge our results and our assumptions. We always have to ask, what are the limitations of what we're doing? It's also not so easy to compare experiments and simulations because of stochasticity, because of randomness. If your parents had had sex on day X in some parallel universe, a different sperm and egg would have gotten together and you wouldn't be here. Even if the same sperm and egg had gotten together, during the course of your development, the particular choices of differentiation, the particular choices of which cells live and die would have been different. And you would have been similar, because you would have been genetically similar, but not the same. Early in embryogenesis, certain genes are turned on and certain genes are turned off, and that's done randomly. So even if you were genetically identical, as far as your parents went, you wouldn't be the same genetically. Your immune system is continually shuffling your genes randomly, certain genes randomly. So even if you were genetically identical in that sense, your immune systems would be different. Your copy in the virtual world would be different. And that makes things difficult to compare. How do you compare two random patterns? If I look at the pattern of blood vessels in major blood vessels, everyone has an aorta. But if I look at the small scale blood vessels that you see, for example, in my wrist or in your legs, those are random. They're not the same in two individuals. If I look in the brain, there are regions of the brain that would be the same in your brain and mine, but they're almost never going to be two cells that would be the same. Once you get past the level of animals like leeches, you can't say this neuron is doing this neuron is doing that. You have clusters of neurons that perform similar functions from individual to individual, but there isn't a homology between the individual neurons. The same thing is going to happen in simulations. If we look at running a simulation of viral infection, each time we run it, we get a different answer. There's similarities in those answers, but we have to think about how we can compare those outputs in the face of randomness. Not easy to do. And how do you compare movies? There's a nice test, which is to look at it and see, does it look similar? But as we found out the hard way many times, things can look similar, but have come from very different causes. And that makes life harder. And there's a lot of scientific information we don't have. RNA interference wasn't understood 20 years ago, and yet it's one of the main ways cells regulate themselves. We don't know that there aren't other biological mechanisms that are in hiding that we don't understand. When we think about things like the immune system, we really don't understand how it works. Coupling between elements is tight. That's more of a technical issue, but makes modeling more difficult. You can't separate things as cleanly as you do in some engineered systems. The range of length scales is large, as I mentioned. There's no complete model. If we can do it, we've talked already about what you could do. Personalized medical treatment, virtual controls, treating diseases before they happen, the ability to build tissues that you could use to replace parts, almost an unlimited number of possibilities if you could get this to work. And it's that vision of what could happen that's kept me in this business for a long time. I don't think it's going to happen during my career, but if any of you go on in this area, I think it could happen in yours. I mentioned a few examples where this kind of modeling is used. in the clinic. It's used, as I say, in our not the particular modeling methodology we'll teach in this class, because they use actually simpler models, more like the ones we did in the fall. Artificial pancreas. There are models of the heart that are used to design heart surgeries. There are models of the brain that are used to design surgeries for epilepsy. Modeling is used at the continuum level to design radiation dosing schedules for cancer treatment and for cancer therapy. And I'm always referring to the wonderful work out of Moffitt Cancer Institute in Tampa, Florida, which is using models that are rather similar to the ones we will do in this class to design better chemotherapies, personalized chemotherapies, in particular for prostate cancer, that have been very, very successful. But it's still a new field. It's an emerging field. And again, the real potential of this will come when you combine it with data-based approaches and AI approaches. So I'm going to skip that one. There's another textbook, another old textbook, single cell-based models in biology and medicine. There are many different ways of representing cells, and each one of them has advantages and disadvantages. Which one you use depends essentially on what kinds of questions you want to ask. If you start out with what are called cellular automata, where you represent cells as points on a lattice, you can represent a lot of cells that way. You can represent 10 to the 7th, 10 to the 8th cells. But there's very little detail, and the range of things they can do is pretty limited. There are the kinds of center models where cells are particles but can move around freely in space. Paul's PhysiCell does things that way. Andy's Mechanica does things that way. And the great advantage of those is that the cells are simple. You can model a lot of them. The models run very fast, but they have limited spatial resolution. The modeling methodology we'll use is on a lattice. The cells have spatial extent, so you can represent a little bit more geometry, but the cost is that you can do fewer cells. And then there are higher level models. For example, you could take lots of center models and cluster them together, just the way you can build molecular dynamics models where you take atoms and combine them into molecules. You can take point-like cellular blobs and clump them together to make cells. That winds up being computationally pretty expensive, but it's quite pretty. And if people play with Mechanica, you could probably do that. And then there are more mechanical models derived from solids modeling, finite element models, which give you a lot of mechanical and spatial detail, but are quite expensive computationally. And so depending on how many cells you need to put into your model and how much detail you want, you might pick different methods. And here are just some pictures of different approaches to modeling. Here are some center models from Dirk Drezdow in Germany, some center models from Sandy Anderson, who used to be in Scotland, is now at Moffitt. These are models that have gone up a scale where it's not individual cells that are modeled, but clusters of cells, blood vessels. This is one from Sasha Popel at Johns Hopkins. He's turned that kind of modeling into a very successful, very well-funded startup company. Big scale, very successful startup. Here is an example of one of these mechanistic models from Karniadakis, does beautiful work on blood flow. These are individual red blood cells. You can see a little bit of texture that represents the tessellation of the triangulation of surfaces of these blood vessels, of these blood cells. There's a huge amount of spatial detail here. These are also extremely expensive computer simulations to run. And so it takes many, many hours of computer time to run the equivalent of simulating of 1/10 of a second of blood flow. This was the model we did, which is sort of intermediate scale, and some other different kinds of modeling methodologies. These are called Vertex Models. This is by Okuda, a very nice Japanese group. These models are very good for modeling bending and folding of sheets of tissue during development. Here's some of Paul's, an older work of Paul from older version of PhysiCell. Biocellian is another center model focusing on very large scale computer simulations, and some other varieties as well. This is from the group that used to mainly be at Oxford. They moved to Australia. They're now in Melbourne. Chased, James Osborne. It's a group we collaborate with quite a bit. So there are many different approaches to doing these things. Morpheus uses the same modeling methodology we do. That's out of Technical University Dresden, very nice software package. Virtual leaf, plant cells are not soft and flexible the way animal cells are. They have rigid cell walls. This was developed by my old student, Roland Merks, who's on the faculty in Leiden, in the Netherlands. And so again, there are many, many options for these. And again, you can read a little bit more about these things. So with that, I'm going to say a few people came on the call to talk about possible project ideas. It may make more sense to people once I've shown some other examples, but it's not fair to make everybody on the, our visitors to the call stay on the call until 8 p.m. to present. So I'm going to give just a slot or two now to do some quick teaser presentations. They can come back next week when you've seen the rest and do it again. So TJ, are you there? Did you want to present quickly?
Yeah, I'm here, James. Thank you. Hello, everyone. I am a postdoc working in James' lab. I've got a couple of slides to maybe just give you some ideas for some projects that I would be happy to invite you to partner with me on. I'll share my screen here real quick. Let's see. Okay. So nothing too formal here. And of course, please just interrupt me if you have any questions. I just wanted to throw a couple of project ideas up, just some things that we've been working on that I would be very happy to have some of you join on. The first project is a new technique that we've developed and have just started to really explore, which is to start with something like a non-spatial ODE model. In this case, shown here on the left, this is just for a simple infection model. They have some virus that's infecting some susceptible cells, and they're moving through a viral life cycle and eventually dying. And what we have developed and are starting to look to apply is to go from this non-spatial model to generating a spatial analog with a CompuCell3D based, a POTS based spatial analog. And that's what you see here on the right. So this is a 2.5 dimensional spatial analog that, when homogenized, then comes back and generates the non-spatial model. Now, this may not look too impressive because this is, a fairly primitive model. But in our first real application where we've done this, as James mentioned a couple of times, with lots of unknowns concerning the immune system. So we have in one of our first big projects with this, we've taken this pretty complicated model of the immune response to influenza infection. This is a non-spatial model developed by some of our friends at the University of Pittsburgh. This was calibrated to mouse model data very well. And our project in this case was to take this system of 20 ordinary differential equations and generate a spatial analog. And that's what you see here on the right. So this is one of the test cases in those simulations. So now for those of you who may have done some previous modeling work with ODE-based models, like the one shown in the previous slide, if you would be interested at all in generating a spatial analog, as you can see here on the right, in this bottom set of subplots, these are comparing spatial model results shown in colors versus the original ODE model solution, which is shown in black. You can see that there are some obvious differences even for a very simple model like this. When we get into more complicated models like something like shown here, the differences become much more drastic, and there are lots of spatial mechanisms that can be investigated that are not available in the ODE models, at least not explicitly. So I'd like to invite anyone, if you have any ODE non-spatial models that you'd like to work on generating a spatial analog for. This is a lot of fun to work on and a really good way to really dive into what is described by non-spatial dynamical expressions. And then the second project, so this is using some of the latest functionality that we've developed and released in CompuCell. So this project is on doing very basic modeling of active transport in polarized epithelial tissue. The most obvious case, to me at least, being in the intestines. So epithelial sheets line a lot of stuff in our bodies, and they do a lot of very interesting things. One of the things that they're very good at is that they're very good at selectively filtering things due to their polarized nature. So in this case, in the in gastronomical processes, they're very good at, say, pulling certain things out of their environment across their epithelial layer, like shown here in these diagrams here. Now with this new functionality that we have for doing transport modeling, diffusive transport in particular, we can now very readily take, say, a model of polarized epithelial layers. So this is a recent model developed here in CompuCell shown on the right. We've got basal surfaces shown in red. These are polarized epithelial cells. Basal is red and apical is blue. And with this new functionality, we can now do things like model active transport across epithelial layers. So if anyone will be interested in, say, starting to develop very basic modeling for active transport in polarized epithelial tissue, please let me know. My email is shown here in the bottom right. So please contact me. I have to go here in a few, but yeah, those are the two projects. James, I'll turn it back to you unless anyone has any questions.
People are free to jump in at any time. You're muted. You come in muted, but that doesn't mean you have to stay muted. I mean, I was thinking, what other thing one could do with this kind of modeling potentially, and I don't know. I see Jeffrey is in the class. Is you could also model the viral infection model. Is a classic case where you go from ordinary differential equations to a spatial model. But another place that's been done a lot is in synchronization of biochemical oscillators. And so one could take a model of, for example, cardiac contraction, where the individual cells were following an ODE model of cardiac contraction, and then see how they interact. Or you could model neurons where the individual cells have neuronal oscillators inside each one, and they interact with their neighbors, and then you get spatial patterning there. So those are some of the kinds of things which you could take. Again, I keep saying that you're not required to have taken the course last semester, and you won't be held to anything from last semester, I promise that. But there are several people in the class who did take the course last semester, and I'm trying to give some ideas here about ways, if you like the project you did last semester, how you could potentially take that project and turn it into a follow-on project. You might also have decided that what you did last semester stayed last semester and you want to do something totally different, which is also fine. But these are some examples of places that one could connect that kind of modeling, this kind of network modeling we did last semester to this semester. And I think perhaps we'll hear a little bit more about that. Is Josh on the call?
Yes, I'm here.
Did you want to present something now about?
Yeah.
I penciled you in for later, but why don't we have you talk now? Because it seems like since you're doing infection modeling, it might be natural to have you talk now.
Right. But in my case, it's just a continuation of what TJ was saying. The model, the project I want to work on is basically the last chapter, what would be the last chapter of my thesis dissertation. And I am happy to have students join if they want to, especially because this is a project that stands by itself, and it could lead to either a Nanohub model that can become a publication, or something a little bit more elaborate. But it's just in terms of the project itself, it's just a continuation of what TJ was explaining in terms of the workflow that we have developed here at the lab. To start from a set of ODEs and develop a special counterpart of those ODE models. Specifically, I'm the problem that I am interested in working on is vital co-infection. It's a project that we are already in collaboration with from a professor from I think it's University of Tennessee, right? So it's an ongoing project. I would be more than happy to have anyone that might be interested in this particular topic to join me. And we have done at this point we have done very preliminary work. We have developed like a dummy version of what a co-infection would look like in an epithelial tissue. And we have done some statistics based on different assumptions about how those co-infection interacts. But we want to be able to develop a more complicated model and to be able to explore more a different set of or more complicated hypotheses.
I mean, it might be worth saying just a word about the biological background. So there was a lot of worry that when the fall and winter came around, that COVID plus influenza would be lethal.
Right.
And what we've discovered is that the same protections that social distancing and mask wearing that protect people from COVID also protect you against influenza. And so the incidence of colds and influenza this year is down enormously compared to normal. So we haven't seen that much co-infection.
Right.
But the worry is the following. There are other respiratory viruses that individually are not very lethal. And influenza, depending on the strain, can be not very lethal or very lethal. But what was noticed by Amber Smith and some of her others is that there are funny immune cross interactions where if you get the other virus first, you don't get the flu, or your flu is very mild. But if you're unlucky enough to get the flu first and get the second virus about 5 days to 9 days later, you die. And the reason seems to be that the influenza, all viruses have to suppress your immune system to some extent to be able to spread. If they suppressed it completely, you'd die from the virus. So the fact that you don't die all the time means that the suppression isn't complete. But sometimes the particular way that a virus, in particular influenza, suppresses interferon response can mean that the second virus that comes in, which would normally be turned off by the interferon response, is able to spread very, very rapidly. And so you can have these very, very serious interactions. And so there was a lot of worry that that might be the case with influenza and COVID. Seems like we may have dodged that bullet, although we don't really have a lot of statistics on it. But this issue of co-infection is quite important and interesting.
Right. And it's not just limited to vital viral co-infection in the sense that you get infected by two different viruses. In the case of influenza-induced pneumonia, people are usually develop other types of infection, especially bacterial infections that accompany serious influenza infections and can be lethal. So the topic, it can be widened. I'm focusing on the viral co-infection because it's meant to be, as I said, a project that can be accomplished within a limited amount of time. But this is an area that people have developed research programs, have done their research, the whole research career around this topic. And it's actually very, has important applications.
And the modeling itself is not very complicated.
Right.
Thinking about it and designing a good problem is hard. Validating against experiment is hard. But the actual software side of it is not too complicated. Okay, great.
Right. Okay. I'm giving it back to you, James.
Again, if there are any questions for TJ or Josh, you could ask now, or you're welcome to talk to them later if their emails are available. So that we'll come back and talk about some other possibilities a little bit later. But I wanted to have them present their ideas. I'd like now to show you some examples. Again, some people have seen this talk many times. About the kinds of questions that we could ask with virtual tissues. One of the things that my group's worked on a lot is neoangiogenesis, the pathological growth of blood vessels. That's a classic phenomenon in cancer, but it also happens during aging in our eyes and in diabetics in our eyes. And I'll just give you a few quick examples of this. What I want to emphasize before we get too far into this is that all of the simulations I'll show we're using CompuCell. And so CompuCell, this software package that we develop, that TJ develops and we work with, is a platform that allows you to build a whole variety of very, very different kinds of simulations. And one of the reasons it's a little bit difficult for me to target this course before I've talked to you and found out what you're interested in is that you could do all sorts of different things. And I want you to do something that's of interest to you. And I can't prejudge what that is. People come sometimes from developmental biology, sometimes from toxicology, sometimes from tissue engineering. And so I want to try to be responsive to that. So let me give you a couple of examples. I invited Gilberto onto this call, but he couldn't make it. He's in Brazil, although he's a long-term collaborator of the group and has spent many years here in the United States. And he was trying to understand crawling of individual cells. If you have a cell that's crawling at a substrate, how does it crawl? And these were models. I mentioned that there are different granularities, different levels of detail that you could build models with. This is an example of building a model of a few cells with a lot of detail. And so in this case, this is a model of a non-polarized cell. You see the nucleus in gray there. In green are what's called the actin cortex. The cells initially are sitting there. The cytoplasm of the cell has been made transparent so you can see what's going on. That cell's sitting there not doing very much. You give it a little bit of a nudge, which you could do in a real experiment. I don't know why my movies aren't going to play. I don't know what happened to that. Well, you're not going to get to see that movie. I'm not going to take the time right now to fix it. I don't know why that movie doesn't want to play for you. That seems to be an interaction with Zoom. As I mentioned today, I've been having trouble with Zoom. It's odd that this one plays and this one doesn't. Let me see if I can get that to play. Sorry, but at some level, it's not that important, but cannot play media. Okay, I don't know. My apologies. I don't know what happened from. Something has happened from this morning till today. Another thing is the work that Maciek and some of the others did, trying to understand cancer progression. And I roughed out a model on this for Nanohub. Having done this work a long time ago and then put it aside, Jeffrey Fox asked me to do a Nanohub demo for it. And I spent my whole Christmas vacation writing a simulation for Nanohub. And I presented it a few weeks ago. It could use some more work, but in cancer, for cells don't go from being normal cells to being cancerous cells all at once. They have to change many aspects of their behavior. And there's a stereotyped sequence of behaviors in which, sequence of steps in which those changes happen. If you look at the literature, you'll see a lot about mutations. However, mutations are not directed. A mutation in a gene could make you make more of the gene or less. They could make the function stronger or weaker. There's no particular reason that a mutation, variation, has a direction to it. And so the fact that you see these changes in cell behavior during cancer has to be due to selection. That is, it has to be due to whether the cell in the environment it finds itself in, which is the extracellular environment, the immune environment, and the cells around it, whether that cell's able to reproduce or not, the cell's fitness. And therefore, cancer progression is fundamentally a disease of selection, of what's called somatic mutation. And so these were some simulations that we did a long, long time ago with Heiko Enderling, who's now a senior professor in Moffitt in Florida, who I mentioned Moffitt before, of this competition just for space between cancer stem cells and somatic cells. And what you see is something that you see in actual cancer, which is that the fraction of stem cells goes up in time during cancer somatic evolution. And so that's a simulation that I packaged up as a little bit of a Nanohub app, but with a little bit more love could be actually a much more interesting simulation. One of the things that a simulation like this shows is that there's a rather paradoxical and very unfortunate effect, which is that most cancer treatments make the cancer worse. If the cancer treatment is completely successful, you eliminate the cancer completely, that's wonderful. But radiation and chemotherapy rarely get rid of all of the cancer. Some of the cancer cells survive, and the cells that survive then tend to become more aggressive and to be resistant to treatment. In particular, in breast cancer, for example, when you have breast cancer recurrences, people will be put on a particular chemotherapy. It'll work for a year or two and then stop working. They'll be put on another chemotherapy, usually one that's a little more toxic. It'll work for a year or two and then stop working. Eventually, they run out of treatments and they die. It's that coevolution due to selection pressure from treatment that makes cancer one of the reasons that cancer is so hard to treat. That's something that one can study in very simple models and build some insight into why that is. And so that's a possible project. This particular model that I did with Heiko published a lot of papers using his own methods on this. This was work that was never published. And as I say, I have a sort of a roughed out version for a Nanohub app. These were the simulations that Maciek and company did. One of the things that cells do in cancer progression is they stick to each other less. which is why they eventually are able to leave the tumor and crawl out and metastasize, form secondary tumors. And this was a simulation that Maciek did showing that when you have a strong immune response, the cells stay connected to each other. They don't begin to migrate. But when there's an immunosuppression, the cells rapidly lose their adhesivity to each other and begin to crawl out and form secondary tumors. That's something that's actually seen in a sort of a tragic way with organ transplants. So when you have an organ transplant, you have to be put on immunosuppressive drugs because you don't want your body to reject the transplant. And it's unfortunately quite frequent that somebody will have a successful kidney transplant, a successful liver transplant, a successful heart transplant, and then develop a lethal cancer very soon afterwards. And the reason is that as you age, you have lots of small tumors in your body that are potentially cancerous, but are held in check with by your immune system. If you downregulate the immune system for some reason, either because of a disease or because of, I'm sorry, somebody's been putting a chat. Okay, that's from because of a disease or because of medicine that has to be used for this purpose, sometimes those tumors are able to break out. And so you have this tragic thing where somebody waits many years for a heart transplant or a liver transplant. They have the transplant, and then they die a year later from a cancer due to the immunotherapy. And so that's the kind of thing one can study with this kind of model. I mentioned Roland Merks, who wrote the virtual leaf simulations. He was a student of mine many years ago here at IU. We worked together on how blood vessels form, how blood vessels organize and form. There's some very interesting processes. Endothelial cells crawl in response to chemical signals in their environment. They reorganize in response to that. And so we were trying to study how that happened. This is an example where I told you that sometimes you can have a simulation that does what you see in the dish, but that isn't right. And it winds up that here, this is actually a movie from a group, actually. The Shada and Preziosi. Ambrosi was also in that group. So this is from the Torino group, doing blood vessel growth in vitro in the lab. We used to do that in my lab too. We actually have a patent on how to do it in the experiment. And this is a computer simulation. These two simulations are both cells that are signaling to each other by chemicals. In one case, that's the cells respond everywhere on their surface. In another, they respond when they're touching the medium, but not when they're touching each other. And that gives you two different geometries. One case you get blobs, in the other case you get vascular trees. And we were able to show in experiments that if you turned off what's called contact inhibition, so that the cells responded everywhere on their surface in the experiment, you also got blobs. And so that seemed to be a pretty good explanation for vasculogenesis. It winds up it's more complicated than that. Biology is always more complicated than that. But that was some simulations we did a very long time ago, and which we used then to build simulations on top of that for the future. I'd like now to take a sort of a break. I try to make these classes not all lecture so that we do things. And so what I'd like everybody to do is do a quick run of CompuCell. People last semester, we did almost all of our simulations using Nanohub Tellurium. Nanohub has a CompuCell, and we'll run CompuCell on Nanohub. The Nanohub CompuCell isn't quite as convenient as the desktop version. And so if you have it desktop, you're welcome to do that. But I'll walk you through the Nanohub CompuCell. The advantage of the Nanohub deployment is that you don't have to install anything on your computer. And in particular, if you have a Windows machine, CompuCell will download and install immediately. There's no problem. If you're a Mac or a Linux machine, Mac, it should install okay, but sometimes there are issues with particular versions of Mac. Linux can be a bit of a bear. But so the advantage of using a web browser interface is that you all get the same thing. So I'd like everybody to do this exercise with me. And I have a poll. People from previous classes will know I'll do a poll, which will say, how are you doing with the exercise? And when you're done with the exercise, you click finished exercise. And that way I know when people are done. So I don't have to keep asking you, are you done? Are you done? Are you done? And if you're having a problem, or I think everybody's done and you're not, click need more time and I'll know. The one thing that I have to say about these polls is you don't get to change your answer. That's an inconvenience of Zoom. And so if everybody immediately clicks need more time, then I have to start the poll again, because the goal of this is to make sure that I give people time to finish the exercise. So sort of give yourself a little time. If you get stuck, by all means, let us know. But don't answer the poll immediately and say, I need more time. Because if you do that, you'll figure it out once you've done it a few times. So let's do some CompuCell on Nanohub, and we'll run that angiogenesis simulation. So the first thing I'll say is that CompuCell is a package, and that package consists of two pieces, a player where you run the simulation and an editor called Twedit. And the editor is nice because it has various features that make it easier to write CompuCell simulations. In particular, almost all the Python code that you'll need is templated in the editor. So if you don't remember how to do a loop in Python, or you don't remember how to use a dictionary entry in Python, that's all in the editor. So you say, I want to reference the cell's dictionary attribute. You pull down a menu, it's there. Once you learn it, you can type it for yourself. But a lot of the things that you'd want to do are templated. It makes it a little bit easier to learn. So let's try to run an angiogenesis simulation. And let me make sure what kind of share I've got. So now I need to go to screen 2. Okay. Okay, so what I would like people to do, I'd asked people by previous email, anybody who took the course last semester, and most people who took other courses related to it will already have a Nanohub account. So is there anybody who does not have a Nanohub account? I'm not hearing an overwhelming set of no's. So if you have a Nanohub account again, please log into Nanohub. If you, for some reason, don't have a Nanohub account, didn't want to say anything, that's okay. You could create the Nanohub account immediately. And what you want to do is search for, actually I have the wrong one here on the screen. I will put into the chat. I'm trying to put into the chat. Maybe I won't. Juliana, do you have the link for base CompuCell?
Give me 2 seconds.
If I try to search it, I'll mess up my screen set.
I guess I have. I pasted to the chat.
That's perfect. So everybody click on that Nanohub tool app. Actually, you could do it from the one that I showed, but it's a little easier to do it from this one. You should get that. And if you haven't created a Nanohub account, or you don't have Nanohub running, when you hit launch tool, it'll ask you to log in or to create an account, and then you have to come back to that location. Okay, and so I'll give people a minute because I don't want to lose everybody. I'll do my polling exercise. I'll launch the poll, and when everybody gets to this window, You should hit launch tool when you get there, but I'm going to wait for a second so everybody can see that search term. It's CC3D base 4X is the tool name. And when you hit launch tool, it may take a little while. It'll pop up a window. Let's see if it's going to work for me. Giuliano, is there a problem? I may have another copy running. That may be the issue.
On yours, I think it crashed because of that red dot. Here, for me, it's still loading.
It's because I have it running already. Huh. Why is it? Today does not seem to be a happy day for me for computing. Oh, I've got it here because it's already open. Okay. It won't let me run it twice at the same time. So when you hit launch, you should see this. And so I can relaunch the poll and see if everybody has gotten to that. And if you haven't gotten to that and you're having trouble with it, somebody says they need help. Giuliano and Josh, can the person who says they need help identify themselves? And Giuliano and Josh, can you see whether you can help out?
Yeah, mine isn't working.
Okay, what's happening?
It's doing the same thing yours did, where I'm just getting a failed to connect to server.
Okay, so try closing it, terminate that session, and then again, as I say, Nanohub is very convenient in some ways. It's a little kludgy in others. If you go up to you kill this session, so you hit terminate there. You go up to here to log in, go to dashboard, and then start it again. So one thing I recommend is that you bookmark. When you find this, when you first do the search, you bookmark this. There's a favorites button and bookmark it so you don't have to search it again. Give that a try, and if it's not working, we can screen share. And I could also set up a breakout room if you need it. I hadn't.
Yeah, weirdly, I got a different error that time.
Okay, try one more time, and then we'll. So it's a little frustrating. It says the whole point of this is to not have this kind of problem happen. So, because it should be completely independent of your computer, since it's since everything is running on their server, if it runs for one of us, it should run for all of us. Is anybody else having trouble getting it running? Okay, Drew, I created a breakout room so I can put you there if you need to do it.
Okay.
Is it still not working?
No. Could it be my browser maybe?
What are you using as a browser?
I was using Opera.
Josh, you're a Mac person, right? Is that an issue?
Shouldn't be. Do you know how to reload a page asking for the cache to be redone?
To like clear my like
Yeah.
cookies and stuff.
Yeah,
Okay.
maybe that's a good idea.
We'll give it one
Actually,
more shot. We'll give it one.
I just tried it in a different browser and I think it's working maybe. What's it supposed to look like?
It should probably give you a blue rectangle for a while.
Yeah. Okay.
Okay. Right. So Giuliano gets the privilege of reporting bugs and annoyances to Nanohub. So if this winds up being an ongoing problem that you can't use a particular browser, Giuliano gets to notify them that they have a problem. Okay. So does everybody then have this window open? So if you come in here now and click on, so you should have seen a blue screen like that. And then we can go through, we'll go through how to work with this. Once it's open, you can resize it. And occasionally there's some issues with getting the sizing to work. And for some reason, this seems to be with Nanohub's X11 implementation. If you have some misdrawing of the main screen, if you pull on the outside of the screen and rejoggle the size a little bit, it'll redraw it and should clean it up. And I will occasionally forget that. All right, so you can, and you have your usual close, full screen, minimize buttons here. This is CompuCell Player that we're, sorry, we're in Twedit here. This is the CompuCell editor that we're in here. And then if we go here, this is Player, where we'll execute simulations. And so you can toggle back and forth between editing a simulation and running it. So now we're going to try to open the simulation. And Giuliano, this is your code, your instruction. So you better help me walk through it properly so I don't break it.
Yep.
Okay. So we're going to go to, we can hit this open project button here. And let's see, I should be doing this in Player rather than Twedit. So really doesn't matter, but I'll full size. So we're in Player. So everybody should go to Player. If people have problems with that, let me know. And we'll go to File and we'll do Open CCD3C Simulation. And we then should see a library, and we need to make sure that we're in the right Glazier is my user space, not in computer. And there should be a bunch of things. This will be slightly different depending on I've uploaded some other things. For example, you see Amber full model. That's the one Josh was talking about. We should have a directory called CompuCell3D demos, and we'll open that. And then let's see, where is it hiding inside of here? It's under demos, isn't it?
Yep.
Demos. And then it is
Book chapter.
Book chapter. Angiogenesis. Select that. That's a lot of directory levels down. So I'll give everybody a chance to try that. And we'll hit open. And you'll see the name here will go to angiogenesis. So let me try that again for people. Where did my poll go? So if you need help finding it, Juliana, we can walk through that hierarchy again. So does everybody have the player running all right? Does anybody not have player running at this point? File, open simulation file, click on your username, click on CompuCell3D demos, click on demos, click on book chapter, and then click on angiogenesis and then select the file. I mean, to some extent, you could run any demo. This is just to have the experience of running some demo, but all run it together. How are people doing in terms of finding it? Mostly people have got it. So. So once you've gotten there, these buttons at the top here, actually the hierarchy is here. So it's displayed. I'll come back to that. If you want to run the simulation, I'll come back to the previous slide in a second. I guess people don't use cassette tapes anymore, so these buttons don't mean anything. What is it called, a retronym, when you have an image that references something like a floppy disk that don't exist anymore? So the button with the arrow on it is play. The button with the circle on it really should mean record, but doesn't mean record, it means step. So that does one step of the simulation and then pauses. The one that's grayed out here is a pause button that lets you halt the simulation temporarily and then restart it. And stop, the square button stops the simulation, and you can't restart. If you start, it'll start over from the beginning. So in general, don't hit stop unless you really want to have to go from the beginning. And you hit play to play the simulation, and we'll see what happens. If people are still working to try to find the simulation, I'll leave it open here. And you'll notice that the screen size got a little bit messed up, so let me try joggling this and see if it helps. It doesn't want to let me do it. There. So you may have to hit pause, and then you can resize. Under window, there's a nice command called tile that will resize the simulation to fit your screen. And you can pull, this console window doesn't do very much. This properties window also doesn't do very much for the moment. And so you can resize your simulation. And you can see already the simulation is actually doing, it's a three-dimensional simulation of capillary endothelial cells moving around, and they're already organizing to form struts, to form blood vessels. And I can run that for a while and see what happens. Pause it. Is the person who was stuck, who said need more time, do you need help or is it working for you now? I'll just relaunch the poll. So I say instead of continually asking you that, I'll relaunch the poll and give you a chance. Again, you can download. I mean, for software development, I think it's more convenient to have CompuCell running on your desktop, probably. Somebody says they have blue screen for player. It may take, if it's still blue, it takes time. If it's blue after 10 minutes, then we have to worry. But if it's blue, it means it's working. If it gives you that black screen and a red circle that says failed, then something else happened. If it seems to be taking a very long time, I would recommend hitting terminate and then launching it again. The advantage of Nanohub is it's a free server. The disadvantage is that it's a free server. So they don't guarantee performance. So if a lot of people are using it at the same time, it can be slow. Your tool crashes when it hits run. How does it crash? Does it throw an error? I suspect that if your tool crashes when you hit run, something that's actually rather important, is that you had to click at the top of your directory hierarchy. You had to click on your name. If at the very top, when I go here, I'm about to stop it to open a new one. Sorry. I'll hit stop. If I go to Twedit and I hit open simulation, and I click on, if I don't click on my name here, and instead I click on demos here, it will crash. So you must click on your username and then click on the version of CC3 demos that's under your username. If I went up here and did, when I started this, it won't let me back probably. When I started it, if I clicked on demos immediately without clicking on my username, it would take you to a version here when it didn't, where it didn't work. So you say you don't have CC3D demos. Giuliano, do we have to do an upload for people? Who was able to get it to work and who wasn't? Was anybody able to get it to work? 6 people said it worked. And 2 people said it didn't work. Kali, you say it doesn't work. If you're saying it doesn't work, that's difficult. So is it's crashing on you when you try to run it? Kali?
Yeah, when I try to click the simulation button to begin the demo, I get the black screen with the message saying that the server has been disconnected.
But so you were able to go, but you had CompuCell3D underscore demos available to you?
Yes, I made it up to that point.
And so you hit run and it crashed on you.
With server termination. Yeah, so because it's server termination, it sounds like it's a Nanohub issue and not CompuCell.
Actually, I was able to get some results, but I need to purge my Nanohub storage to simulate. Maybe for you, Kelly, maybe it was full for your storage, and then maybe it's crashing. I'm not sure.
That could be. And
I mean, the only thing I can, and you tried, I guess the only thing I can suggest is terminate the application and relaunch it.
Yep.
And if you've tried that already, I'm trying to think what else I can suggest.
And David, I don't know how it wouldn't be there. Are you sure you opened the CompuCell main tool before?
How did you get to CompuCell?
Yeah, I got to CompuCell through Nanohub, but I also had it open on my computer as well.
Well, there are several CompuCells on Nanohub. That's the thing.
Right. So you're saying you have it running on your desktop as well?
Yeah, just the player.
Okay, but that's all right. So if you have CompuCell Player running on your desktop, you should be able to do the same thing without having it. So then you shouldn't have this issue. So if you run it in your desktop machine, you should be able to go to demos and run it.
Okay, I'm trying that now.
Usually it's the reverse. Usually people have trouble with the desktop app, which is why we went to Nanohub. But as a backup today, if it's not working on Nanohub, we'll use the desktop app. Is anybody else stuck? Kali, are you still stuck?
Yeah, I'm still stuck.
And it's still giving you the same, what was the error it was giving?
The error specifically was, sorry, I'll type it in the chat.
Everybody else, so it looks like most people have got it working, but so if you don't have to, while we're debugging Kali and other people, I encourage you to play with CompuCell. One of the things you can do here is that there is the cell field, which is showing the cells. And if you go here, there's a chemical field, which is the vascular growth factor. And you can switch and visualize the chemical field. When you switch the visualization, you'll have to hit run again. It'll pause. You can also do a 2D slice through your image, which may be more informative when you're looking at chemical fields. Run that. And so you can play around a little bit with how visualization works. If you want to see both the cells and the chemical field at the same time, you can hit this new graphics window button here. And then under window, a very useful thing is this tile function that lays out the images for you. And so I could, for example, visualize my cells in 3 dimensions. And then I can zoom with my the roller on my mouse, rotate my image to be what I want, and I can let that run. So why don't you play getting familiar with the controls in CompuCell? If it breaks and dies on you, you can either restart the simulation, or if player goes poof, you go back to the Nanohub dashboard and start again. Server disconnect, target closed. That one, I'm sorry, Kali, I do not know. I do not know what that one is doing.
Yeah, the only time I've seen that one was when I had too many sessions open or when Nanohub was acting up.
Did anybody else have that particular error? We normally take a bit of a break in the midpoint, and believe it or not, we're only more or less at the midpoint of the class. Does anybody else need help, or does everybody else have it running now? Anybody else need help? So why don't we take a break? Kali, you may lose your break. to this. Why don't we, do you want to screen share to us so we can look at it? Okay. And on the break, why don't we say we'll start back at 6:55, give you a 10-minute break. Okay, so yeah, try launch tool. Oh, so first, before you launch the tool, go to your dashboard. Go up to the logged in dashboard. Okay, so you don't have anything running. You have plenty of storage. Okay. So now let's do CompuCell3D main tool on the right, there. You can just click, okay, launch tool. Is that doing anything? Because it depending on how you've got the Zoom screen shared, we just see launch tool. We don't see the CompuCell.
It's loading.
Okay, so you'd have to reshare so we can see it.
Well, it hasn't loaded yet. It's just in the process.
Oh, it hasn't done it. Shouldn't take that long. I wonder why. Doesn't usually.
Okay, here we are. Can you see that?
So now you've got a blue screen, which is good. Sort of ironic that blue screen is good.
Yep.
So it's going to take taking itself a while. So it's going to run. Okay. So why don't you resize it? See? Yeah, see, pull. Okay. And let's open it again from scratch. So go to File, Open Simulation. Okay. Wait a sec. Go to click on your. I'll go to go to File, Open Simulation. Click on your name. No, don't click there. Click there. There you go. CompuCell3D demos. Book chapter, Angiogenesis. Yeah, that's sort of a trap, which is that if you click on it, it's there, but it's not the right version. Okay. And now, yeah, there you go.
Now it's going.
Yep.
And so if you go under Window and hit Tile, it'll resize. And if you go where it says XY there, go to 3D. Okay, and now you can, if you have a roller on your mouse, that'll zoom in and out, depending on.
A little too much zooming.
There is, I think, K or F. Reset
Control R
the
is
camera.
reset. Yeah, there's a reset for the.
Oops.
Well, I think, yeah, that solved the issue, though. I was going to the wrong file system.
Yeah, that's unfortunate. The reason for that is that the CompuCell application has to have copies of things, which it then copies into your local directory. But we haven't figured out how to hide that, so that there's remember, this was a problem with Tellurium, that when you launched Tellurium the first time, you could have this non-edit window. It's the same kind of issue with the way they structure things. So there's an edit. It makes an editable copy of itself, but it doesn't hide that original directory.
Mm-hmm.
So if you don't click through to your name then you're in a non-editable directory. Then when you launch it, it tries to write to the root directory, which it can't do. Simple, I guess you said you're having trouble getting it to work. What's happening with you? It's not working.
I'm trying, but it's
How is it failing?
I'm able to open the angiogenesis, but then nothing is happening. I'm not able to come to the window like you have. It's not opening the simulation window. It's staying with the player.
So so you see this window, but nothing inside of it.
Yeah, I mean, this one is not opening.
So you go to File and you go down the file hierarchy and hit Open. And then what happens? Do you want to screen share for a second and show us what you're getting?
I don't know why the camera is not working today with the Zoom. My laptop.
You don't need a camera to screen share.
Okay.
So if you go under Zoom, you should have a share. Okay. Okay, fine. So it looks like it's launching. Can you? Okay. And it's just sitting like that. It's not doing anything else?
No, I just tried it again to launch again just to make sure. I mean, I've done this like 7 times now.
Okay, well, I'm sorry. I'm just, it's frustrating, but once everybody's done it once, it usually works. So it's a
So if you see here, like.
Right, but it hasn't actually, so the player has not yet actually launched.
Yeah.
So I wonder why it's so slow. Why don't you hit terminate. Okay. Go to hit this. Oops.
Yeah, this one.
Hit this button here.
Okay.
Okay. Okay, let's see if it launches properly. Huh. Giuliano, is this familiar to you?
Not for this long.
Don't understand.
I mean, yeah, it doesn't have any of the narrow chord. Oh,
There it goes.
and
Okay, fine.
by the way, simply you can resize the blue window on the lower right so that you don't have to keep scrolling up and down. On that,
here.
that 24 times
all the way at the bottom, at the very bottom corner. Sorry. Yeah.
No, below that, 124. Yeah.
That corner.
And you can drag that around.
Okay.
And now you can.
So I go to File.
Okay. Hit File, yes. Go to File.
Like
open, yes.
Then I go here.
Yes, all right.
And this
here.
one.
Yes, that one, yes.
And
Okay.
then.
Yes.
And then this
Book
one.
chapter, yes.
Angiogenesis.
Yes. And then this one. Yes, that's right. And now hit Run. No, Run.
Run is.
That blue button, Run.
Oh, there. Okay.
Okay.
So I'm getting this.
Right, that's what you want. So now full screen it, hit this button. And now go to Windows here, Window. Pull this down here.
Yeah.
And hit Tile. Tile. Okay, and now here, go instead of X, Y,
Mm-hmm.
go to 3D. And now hit Run again because it'll pause. There you go.
Okay.
Okay,
Yeah, got it. Thank
okay,
you.
great. Okay,
Thank you.
anybody else have any issues? I know the first time you do these things, it's always frustrating, but once you've got it running, it's going to be the same thing each time. So it should be okay, I hope. Okay, good. So let's come back to all right, why? All right, I'm switching. All right. So let's keep going. So we talked about how to start, stop, simulation. I hope you got to explore a little bit about how to visualize things. There are lots of options, really a lot of options. And one of the emails I gave you walked you through all of the directory trees of all the options there are in Player. There are a lot of, you could change the colors, you could change the interval between renders, you could change all sorts of things. And you'll get to explore those. Lowercase r is reset. So let's see if that works. R will reset the image. So if you get things out of field of view, this kind of thing, R, lowercase r, hover over lowercase r. And you could change colors. You could change, if you go here, this little, it's a wrench and a wrench and a screwdriver. It will allow some settings to be changed. The visualization lets other settings be changed. I'm not going to take the time now to go through all of those, but as time goes on, they'll be in the slide deck. They're in the handouts, and you can explore. In general, when you change the rendering, it will pause and you have to hit start again to see what's going on. So for example, let's just quickly, suppose I wanted my cells to, in this case, they're blue. Suppose I want them to be green. I hit the colors here. I look at the cell colors. Currently they're blue. And I want to change the color. I got to make them burgundy. All right, why is that not? I crashed my simulation. Huh. All right, that's a different one. It restarted the simulation. Well, that one's on. That having I outsmarted myself on that one. I don't know what happened there. It's running. Not letting me do a resize. There we go. So if it gets stuck, a resize will help. Let's see if I can get the color to change. Pause. Okay, here we are. Pick color. Okay. Okay. I don't know why that CC3D preferences isn't doing its job. Should disappear. Yeah. Okay, well, life doesn't seem to want to be cooperative today, so I'll let that one go. We'll figure out about colors later. Okay. I already showed you that you could see the chemical field as well as the cells. Then we talked about, I'll give you just quickly some other examples. We already saw this vascular tumor example. That one also lives in the demos. It takes a little bit longer to run. This is a simulation where you have blood vessels that are supplying oxygen and nutrient to the tumor. The tumor is growing. At first, all the cells of the tumor, which are green, have enough oxygen and nutrient to proliferate. As they grow, the cells in the center begin to die because they don't have enough oxygen and nutrient. They send out a signal that causes the blood vessels to grow. That's the purple. And then there's an interaction between the growing blood vessels and the tumor. That's an old simulation, but a quite pretty one. And you can analyze the different phases of tumor growth. There are quite a few, there are 6, and quantify how the interaction between the tumor and the blood vessel changes the growth of the tumor in terms of size, shape, invasiveness, and so on. I mentioned that one of the things we did a lot of work on was early embryonic development. We saw that experiment with a chicken embryo here. Again, I don't understand why these movies don't want to play. They were playing earlier today. They don't want to play now. We can come back to that, I guess. But we can, this is another simulation showing how the cells rearrange during embryogenesis. I mentioned that blood vessels in the retina cause problems of various kinds. Age-related macular degeneration, I hope nobody in the class is having this problem. Age-related, although it does occasionally happen to young people after severe infections. It's one of the main causes of blindness in older individuals. And in this disease, the blood vessels that normally are behind the retina, providing it with oxygen and glucose, cross the barrier that normally separates them from the photoreceptors and start spreading within the photoreceptors and chewing it up. This is a simulation of 3D simulation. See, some of the movies are working, so I really don't understand what the issue is. This is a simulation of the blood vessels in purple behind the retina. The gray region is something called Brooks membrane, which is a thick membrane that separates the blood vessels from the photoreceptors. The green cells are the pigmented retinal epithelial cells, the black lining of the eye. And then the gray correspond to the photoreceptors. And what you're seeing here is that the purple cells have spread into the retina. They've made a hole in Brooks membrane. Once they've gotten into the retina, they spread and they proliferate. They eventually cause the detachment of the retina from its support. And then eventually they'll cause the breakup of the photoreceptors and the death of the photoreceptors. And that's something that happens in a certain fraction of older individuals. It can lead to blindness over a period of about a year from the time it starts. There's the failure of the retina. And one of the things we were interested in is what phenomena in terms of dysfunction at the cellular level can cause this kind of thing to occur. And this was what we call a risk map, where in the normal retina here on the right, you don't have invasion by the blood vessels. And then for various problems at the cell level, impaired adhesion between the pigmented retinal epithelium and the membrane underneath, impaired adhesion between the photoreceptors and the pigmented retinal cells, impaired adhesion between the pigmented retinal cells, all of which occur in various kinds of pathologies. they give you different probabilities of having this condition occur. This is a probabilistic, it's a stochastic outcome. And so the best you could do is get a map of the probabilities of things happening. Remember, I'd asked earlier, how do you deal with things that are probabilistic rather than deterministic? This is one way of handling that. And that actually winds up being something that the simulations predicted the mechanism before it had been observed in the experiment. We've worked a lot with a surgeon in Indianapolis who works on a relatively rare disease called polycystic kidney disease. This is a disease in which the kidneys develop cysts and keep growing until you die. It's a sort of a high yuck factor disease. Typically, by the time you die, your kidneys are the size of watermelons. It happens to be associated with mutations in two genes, polycystin 1 and polycystin 2, but exactly how that mutation translated into the cystic phenotype wasn't understood. This won't play. Again, I don't know why these aren't playing. But these are simulations which we did of an in vitro model of cystogenesis, which was developed by our collaborator, who showed that if you put in a particular cell adhesion molecule into these cells, you got cysts. We were able to replicate that phenotype in the simulations. And then working with our collaborators in Brazil, who were on sabbatical here at the time, we were able to identify, based on that observation, potential drug targets. And Bob Bacalhau, the MD, is currently doing animal studies, hopefully before human clinical trials of these drugs to try to treat this disease. At the moment, you die if you don't get a kidney transplant. There's no drug treatments for it. We've been interested in diabetic retinopathy, another vascular disease of the retina. In this case, the blood vessels, if you have diabetes, as you age, you always lose some blood vessels in your retina. It's normal. It's not great. You still lose some vision, but not a lot. In diabetics, the blood vessels, this is a normal retina on the left. In diabetics, you see the blood vessels get squiggly. That's associated with high levels of vascular growth factors, abnormally high levels of growth factors. It happens in cancers as well. And you also get them to be leaky, which also happens in cancer. And some of them drop out, they disappear. And when that happens, you lose your vision. And so diabetic vision loss is pretty common and pretty serious. Again, it can happen very rapidly. The main way that's treated now is by saying this is happening because the oxygen levels required are too high. And so they go in with the laser and they kill the photoreceptors so they don't need oxygen. This is like fighting a forest fire by cutting the trees down, or fighting a fire in the city by demolishing the houses. Well, it works, but it's also very damaging because you're actually destroying the thing you want to save. And the way it's done at the moment, they go in and they laser ablate. Each one of these light gray colors is a place where they've coated and killed the photoreceptors. If you look at this retina, they've killed a lot of photoreceptors, which is not so great. And so we built a model of the feedback loop that we believed caused this failure of the retina, and then used patient-derived images to try to calculate what the oxygen field would be for the patient, and then specifically tried to calculate what the probability, if a blood vessel failed, you can't say which ones are going to fail, but if a particular vessel fails, what's the probability that it'll cause the neighbors to fail? So basically, if your house catches on fire, what's the probability it's going to cause your neighbor's house to catch on fire? And then based on that, you can then say, well, if you're going to kill the photoreceptors, you should kill the photoreceptors that are likely to cause problems instead of killing randomly. And you can then try to optimize the treatment by saying you're going to kill the photoreceptors where they're risky and not do it where they're less risky. And that minimizes the probability of the disease spreading, and also minimizes the damage that you're getting from the treatment itself. And that's something that we would love to work on more. The student who was working on that was very good. They graduated. They're now in London at the Crick Institute. And so it's a problem that I would love to see worked on more. We actually have a patent on it in terms of treatment. So we'd love to tool that up and get that to be something which could actually be rolled out as a clinical treatment. We've worked a lot on liver toxicity. I'm going to try to shorten things down because we're going to run out of time. Particular Tylenol, acetaminophen. Tylenol, of course, very common painkiller. It's used medically indicated because it doesn't cause bleeding as much as some of the other painkillers. But it has the nasty effect that you can take it safely for years. You can take it then once and die from it through liver failure. And the reasons that it causes liver, it's not well known that it can kill people. The classic person that it kills is a fraternity, a young male fraternity student. And the reason for that is the following. Tylenol causes liver failure by depleting a particular molecule called glutathione, which is a protective molecule in cells. When you haven't eaten anything, your glutathione levels drop. When you've had alcohol, the level, Tylenol itself isn't very toxic. There is an enzyme in your liver that turns it into something that's toxic. When you drink alcohol, the level of that enzyme goes up and the level of the protective molecule goes down. And so if you wake up in the morning having drunk a lot and not eaten, and you take Tylenol for the hangover, and you take 4 Tylenol or 8 Tylenol instead of 2, which is normally pretty safe. Most medicines you can buy over the counter, you could take 10 of them, it won't hurt you. You can die. And so one of the most common ways that Tylenol would be taken, which is a headache for a hangover when you wake up in the morning, is extremely dangerous. And so you have 18-year-olds or 20-year-olds who die from Tylenol. And so we were trying to understand the mechanisms by which that happens. And it really, a lot of people die from it, really a lot. It's no longer true because of the opioid epidemic. But until the opioid epidemic, more people died from Tylenol poisoning every year in the United States than from all illegal drugs put together. It was really bad. It's still toxic, but now unfortunately, there are other things that are worse. And one of the things that's sort of surprising is that if I cut out 80% of your liver surgically, it'll grow back and you'll be fine. And so one of the questions that we wanted to understand is why the damage from Tylenol caused a situation where the liver couldn't recover. And so we did a series of simulations, starting with the Environmental Protection Agency and then working with National Institutes of Health, modeling chemical toxicity in the liver. And one of the things that, and in this case, we were actually able to match the levels of the various chemicals pretty accurately to what was observed clinically in the blood to try to calibrate the model. The hope with a model like this is you start with Tylenol, then you move to other drugs. Many medications under development fail because they wind up being toxic to the liver. So there are a lot of drugs that work in terms of treating the disease, but can't be used because they cause failure of the liver. And so if we could understand what causes liver failure for particular molecules, we might be able to rescue them and get them back into use as drugs, as therapies. And so that's what that kind of use is about. Another question you can ask is what's called patient variability. As I mentioned, some people take Tylenol very safely. Other people have even very low doses can be very toxic. And you could use this kind of modeling to explore why some people are so susceptible to toxicity in this way, and others are very resistant to it. I mentioned that we've done some work on infectious diseases. This is, as I say, a model that was partly started by 2 students in the class last year, Sam Heaps and Kira Breithaupt. Sam was a master's student in the class. Kira was an undergraduate in the class. And they wanted to work, the class decided they wanted to work as a group on modeling COVID. And so over spring break, we coded something up together. This paper has been published now in PLOS Computational Biology. Like the work Paul's been doing, it turned into a rather large-scale collaboration. TJ, you recognize Josh is on the call. Juliana is on the call. Lutz Brusch is the person who developed Morpheus in Germany. We're working with him. Jessica and James Osborne are the people who are Chased people, Chase software package in Melbourne, Australia. Ellen Quardakis is experimental biologist here at IU. Richard Plemper is a drug developer, develops antivirals a little bit like remdesivir at Georgia State University, and that's me. And so we were trying to build a very simplified model of the immune response to COVID. The complexity of the system is very high, so it's not something that's trivial to do. We started out by looking at a small patch of epithelial tissue, lung tissue. We assumed that there was what's called primary response, primary infection. That is, it's a disease you've never had before. You don't have a pre-existing immune response to it. And it's an acute infection. We're looking at what happens over a period of days or weeks after infection, not long term. And you have to model the cells that are being infected. You have to model the virus. You have to model the immune signals. You have to model the immune cells that come in to the lung. And actually, if TJ had not wanted to speak earlier, this really belonged before TJ's presentation, because TJ's presentation was a much more detailed immune model, had many immune cell types and many of the biochemical signals. But a simple model looks like this. You have an initial infected cell in the middle. That infected cell starts producing virus. The virus spreads, infects the neighboring cells. Eventually, the infected cells die. They cause immune cells to come in. And the viral load goes up and eventually comes down. Cytokines are the immune signals that the immune system uses to amplify signals to say there's an infection or to suppress immune response after infection. Oxidative agent, immune cells can kill in a variety of ways, and so those are modeled. And you can then do simulations of the spread of the virus, the cytokine signal, what fraction of cells die in a given period of time, whether any cells survive. And those are a whole series of simulations that one can do under different circumstances. And you can then also simulate, for example, and I'll jump ahead because I want to have time for Giuliano and Andy to present their project ideas too. You can ask the question, what happens with a drug like remdesivir? Now, remdesivir, which is used, if you get sick with COVID, seriously sick with COVID, and you're put in Bloomington Hospital, they'll give you remdesivir. So it's used quite widely. There are now a lot of other drugs they'll give you. They'll give you immunosuppressants like dexamethasone. If you're lucky, they'll give you antibody therapies, although those are harder to get. But remdesivir works, from the point of view of modeling, it's very nice because it works by inhibiting a very specific step of the viral life cycle. It works by inhibiting the replication of the viral RNA. And again, another time we can come back and go in more detail. And one of the questions for this course is if you'd think that you would like the course, you would learn the same things in terms of CompuCell, but they can be taught around a cancer model, which is how we used to do it until last year, or they can be taught around this model of viral infection. You'll learn exactly the same things because the modules in CompuCell are the same, but the deployment is different. You might want to think which of those two is more appealing to you. Maybe the infection model is more fun at the moment. Again, you can always come back and do the other one. But in any case, remdesivir works by inhibiting viral replication. And the specific aspect of viral replication is RNA synthesis. It blocks RNA duplication. And so you can then simulate what happens if you come in and at a given time in the infection, you give a dose of remdesivir, and that reduces the rate of viral replication. And you can ask the question, what happens? And these are a whole series of simulations. Each one of these little green lines represents a different simulation. And you'll see if I zoomed in here, for example, that sometimes this line goes down, horizontal line, this line comes across and goes down. And this is number of uninfected cells. So in this case, all the cells in the simulation got infected. And sometimes a few cells get infected, but most of them don't. So this would represent a case here where the drug controlled the infection. And in this case, the drug did not control the infection. Green here means that most of the time or all the time the virus was controlled. Red means most of the time or all of the time the virus was not controlled. What are the axes? The time of application, when you start treatment, on the horizontal axis. The vertical axis is how effective the treatment is. Top is most effective, bottom is less. So basically the vertical axis is dosage, and the horizontal axis is time of first treatment. If I treat very early on, say before you get sick, and I give you a lot of the drug, the virus never replicates, so I always survive. If I wait till everything's died before I start treating and I give you only a little bit of the drug, it doesn't do any good. It's not surprising. What's interesting is this intermediate region. And one of the big problems with antivirals, especially for things like Tamiflu for flu, is that by the time you feel sick, the virus is almost done. The maximal amount of virus in your body happens just about the time you start getting the headache and feeling crappy with flu. With COVID, it takes a little bit longer for the viral load to maximize. But essentially, by the time you get into a hospital and start getting remdesivir, your body's already reducing the amount of virus using, your immune response is reducing the amount of virus. Now, in the case of COVID, the virus may be spreading. It may start in your nose and your throat and move into your lungs. And so remdesivir may prevent that further spread, and that's very useful. It saves lives. But it's not going to stop the virus from starting in the first place. If you knew who was going to get sick from COVID and gave the drug beforehand, then that would be useful. And Tamiflu is usually used that way. If you go to the doctor and you say you've got the flu, they typically won't give you Tamiflu because it's too late. On the other hand, if you're a parent and you have a child who comes home from school with the flu and you know you've been exposed, if you start taking Tamiflu right then, before you get sick, it works pretty well. And so potentially you could use a drug like remdesivir prophylactically when you know you've been exposed but haven't gotten sick yet, and then you'd expect it to work very well. The problem with remdesivir at the moment is that it has to be injected. And Giuliano will tell us a little bit more about remdesivir as a possible project idea, perhaps, if he feels like it a little bit later. But this is basically an outline where if you give a low, even a very low dose, if you give it early enough, is very effective. Even a very high dose, if you give it too late, doesn't work. And the boundary between these two tells you something about the utility profile of the drug. And we'd like to do this not only for remdesivir, but for things like dexamethasone, antibody treatments, and so on. Okay. This kind of model is oversimplified, and we're working on a whole variety of extensions for that. This is a model of interferon response that Josh is working on with a group at Pittsburgh. They're just finishing a paper on that. You heard about spatialization of models. People who took this class last semester and did non-spatial models potentially have the ability then to take those and put them into this spatial context and see what happens. Josh already talked about looking at competition between viruses. Hey, Furkan, speak up.
Yeah, so actually I have a question about those specialization for non-spatial ODs.
Yeah.
Is it a kind of like stochastic process, or like do you see all same results, or is it a stochastic approach?
So these simulations are intrinsically stochastic. That was a very good question. And so each time you run the simulation, you're going to get slightly different answers. Depending on how big the simulation is and which elements you're spatializing, the stochasticity may be very different. As you'll remember, if you do chemical reactions, if the number of molecules is 10, then the fluctuations are huge. If the number of molecules is 1 million, the fluctuations are small. In the case of viruses, because initially, typically the number of virus particles you're infected with is fairly few, there's a lot of stochasticity in the early time evolution of the virus, and that gets amplified as you go later. The initial immune response can also be stochastic. And so when you run these simulations, Paul's or mine, you'll see that you'll get some replicas where the virus doesn't take, some replicas where it spreads very quickly, and then you'll get sort of a typical behavior in between those two. And as you change the parameters, the frequency with which you get those outcomes will change, but you still will typically get that range of behaviors. At some point, the virus doesn't spread at all, so you never get a sweep by the virus. At some point, the virus always sweeps, but there's a big intermediate regime where you get all of those behaviors with different frequencies. And so these are definitely cases where the ODs are not reflecting the stochasticity of the original model. Now you can build non-spatial stochastic models. That's the way you can run Gillespie you can do that too. The question is whether those reflect the real stochasticity of the problem. Sometimes they do that just fine. If the stochasticity, for example, is the rate of replication of the virus inside the individual cells, then you don't need the spatialization. But one of the things that happens is that if the virus doesn't infect cells in a salt and pepper way, typically what you get are lesions where you get a plaque where a clump of cells get infected and then the cells around them die and you get a circular spreading lesion. That spatial structuring isn't something that you can include easily in a non-spatial model.
James, and if I might add as well, I think the beauty of the implementing this specialization in a systematic way is that you can actually show the conditions in which the parameters, even accounting for the stochasticity, can on average deliver the same results as the, let's say, the OD system, because the assumptions are similar. Yeah. And then you can also show that as you deviate away from those assumptions, then any difference between, for example, the prediction of your OD model versus the predictions of your spatial model can be attributed to the differences in the values of those parameters.
Thank you. I have to say I've been rushing a bit because, as always, it takes time to get everybody to be able to run CompuCell, and then I had planned to. You triple the amount of time you think these things will take, and then you should triple it again. So, but it's important that everybody will run. And so there's some of these issues. This is a plaque simulation that Josh has just finished up with a group at Pittsburgh, studying the growth of these lesions, and how the parameters in the model affect that growth, in particular how interferon affects the growth of plaques. Not something that one could develop further. This is the simulation that TJ showed us. I don't want to talk about his work when he was there to talk about it earlier. Something that we started on that we haven't had a chance to work on very much is some work that was being done by a former student of mine, which is trying to model the formation of lesions in the whole lung. And so now, instead of having our little CompuCell cells representing individual cells, they're representing volumes of the lung. And this is a geometric model of the whole lung and the spread of the virus in the whole lung, being done by Giulio Belmonte in conjunction with Amber. So with that, I was hoping, we'll see how time goes. I was hoping we could run that simulation on Nanohub. But I wanted to give a little bit of time to the two other people who were on the call. Is Andy still on the call? Andy? Andy, you've been very patient. Giuliano, do you want to talk first beforehand? Did you want to say something before about possible project?
I can go after Andy.
Why don't you go first? Because we'll stick with CompuCell first.
Okay.
I mean, if you don't want to, it's fine. It's up to you.
No,
Okay.
So as James mentioned, I also have some stuff with remdesivir, but I didn't prepare a slide for that. But here I have one proposal of collective migration and growth for wound healing. So whenever you have a wound, it's going to close up, right? And to do that, your cells need to replicate themselves, but not too much. Otherwise, you're going to get cancer. And but only growth is not enough. They actually need to become motile, which the skin cells are not, and move about. And they actually coordinate their movement. So this is a possible project exploring how they coordinate their movement. This is using Boids, which is a type using concepts from Boids, which are bird-like particles. So here's a movie of Boids. They're trying to stick together and trying to avoid the white balls. And so here's one occasion that could do that for a cell. It evaluates its own velocity, its neighbor's velocity, and some noise. And besides this, there's the remdesivir project that James mentioned. And I'm also doing some cellularization of ODE's works, but it's on a ODE system for the lymph node, some immune cells from the lymph node into an infection site, and later immune response after 7-10 days of infection.
And I want to emphasize that there's no obligation for anybody to work with any of these projects that you heard proposed. You're free to work on your own on a project that is interesting to you. You're free to team up and work on projects that are unrelated to the ones that have been discussed. But I wanted to let you know what was being done in the group so that if there was a project that appealed to you, you had the option of having some extra help that way. We will all work to help you on your projects, whatever you choose. But if somebody wants to bootstrap by working at a project that's already partly going, you're welcome to do that. And so I wanted to give you some idea of some of the possibilities. again, I sent out a long list of possible projects in the homework, and there are many more, because as you saw from the list of demonstrations that I did, you could do a lot of things with this kind of modeling. And so that selection can be a little bit overwhelming. On the other hand, it can also be very, it means that if you have a predefined interest, we should be able to find something that is appealing to you, I hope. The second person, the second group of possible projects, and this is something that was really, in a sense, directed to Furkan more than anything else, is that because Furkan did this course before, and is back for Latin for punishment, is back for more, and so this course will be about CompuCell, and CompuCell is a very well developed software package. As you see, all these things that we've shown, there are dozens and dozens and dozens of papers by groups worldwide using it, but it does have some limitations. And I mentioned that, for example, PhysiCell is able to simulate a lot more cells than CompuCell because it has a simpler representation of each cell. It's also what's called off-lattice that has some real advantages when you're doing simulations of mechanical deformations of tissues. And so I wanted to give the floor to Andy Samoji. Andy has developed the center model package called Mechanica, that in some ways is rather similar to PhysiCell, with one big difference being that it's Python scripted. And that means that if you want to write simulations, you don't have to learn C++. Not that learning C++ and compiling code is a big deal. You can writing PhysiCell simulations, I'm sure Furkan could show us how to do that. It's not so bad. But Andy has been working on trying to think about ways of making simulations as simple as possible for people who are not computer savvy. And so I wanted to give Andy the floor and talk a little bit about some of the things he's doing, just to give you a sense of maybe not, well, you're welcome to talk to him in this course. So the one, I'll just turn it over to Andy. Go ahead, Andy.
Hi. Okay, so I'm Andy Shomaji. I'm a research scientist here, and I work on a new simulation package called Mechanica. And let me go ahead and share the screen here. I got a couple, 3 or 4 little slides here. So, and let's see, share. Okay. So the goals of our new package here are first and foremost, we're looking at simplicity and ease of use. So we start from the user API first and see what kinds of concepts can we represent, and then what kind of physics can we build using these terms. So it's a pure Python API. The typical models are, I don't know, about 20 to 30 lines of Python. The other big goal is real-time 3D interactive performance. My background is real-time 3D simulation, so that's really my big goal. With about 300,000 objects, we can get roughly 30 frames a second or so. And what we're looking at is actually having users interact in real time with the running simulation. Then also we want to be able to do really expressive physics. So we have concepts such as fluids, chemistry, solids, solid fluid transitions, and the big thing that we do is we do, much like CompuCell does, we have a concept of agency, where users write code for individual objects, and then they specify how these individual objects interact with their neighbors and their environment. And we're looking at modularity and compatibility with other software. So it's a standalone Python package. You write standard Python scripts. And we have both a web-based and desktop-based front ends. So the kinds of problems we're looking at doing are, if we're looking at a single cell level, we're looking at allowing users to create little dash spots and springs and other sorts of mechanical interactions between the elements of a single cell. So we're looking at trying to enable these kinds of really detailed single cell models. One of the projects I'm working on with a group at Purdue is modeling an epithelial cell where we have a mesh of actin cells along the bottom of a yolk, and those are individual objects that are connected by a mesh that interact with a fluid-like blob on the surface. One thing I'm currently working on is a fluid-solid interaction of basically verifying the mechanics of our fluid model and how that adheres to solid surfaces in the presence of gravity, where we can rotate that surface and have the blob slide off it. Things like cyanobacterial filaments. One of the big things I'm starting to do right now is viscoelastic fluids. So how these viscoelastic materials form and create bonds as they move along on a physical object. And also, for those of you more interested on the chemistry side of things, doing things like transport in flux networks, transport in pipe networks, and of course fluids. Really simple Python API. So let me go ahead and run a little demo here. So I'll go ahead and run the demo first. Okay, so this model only does a secrete and a diffusion, but we can see that it runs in real time, and that's just a chemical model. But the API for that, or the model script for that, this is the entirety of that model script, where we create a couple classes to represent our different cell types. We attach a chemical species to them, set its color, write a little function of what of how the cells can interact with their environment, and hook it up with an event, and then that's basically it for that kind of model. We can do more of the mechanical type models. And this is a simple the Pibble model. And that's a quasi-viscoelastic fluid on top of a yolk sac that's being pulled by a mesh of these actin filaments on the bottom. And this is the desktop version we have working. And then we have an early prototype of the exact same code running on Nanohub. And then we can run a simple model where we can create a cuboid there and use a Jupyter image widget to display it. And then we can rotate this guy around. And so forth. As you can see, the widgets and all those other user interaction components are not really well developed on the Jupyter version. And that is one of the possible projects that I think would be really interesting. So one of the possible projects that I have in mind would be if you're more of a software / web type person, I think it would be really great to develop a Jupyter widget, possibly based on the image widget, that grabs rendered images from the simulator as it's running and then displays those in the browser. Another possible project is to explore the rules for fluids and viscoelastic materials, and explore the rules of how we can have the material flowing along the surface and what kind of rules do we need to form and break bonds to create a realistic viscoelastic material. Possibly investigate a Boyd's model with secretion and uptake with its surroundings. Or I have another researcher in UK that I'm working with on creating filament models for cyanobacteria. So that was a real quick rundown of what kinds of physical models we can do and what are some possible projects. So Mechanica, like I said, it's a very new package. It's obviously not as well developed as the more mature ones, but it's written from the ground up with these ideas in mind of real-time performance, expressive physics, and ease of use for the user. So, are there any areas there that we might have some interest in, or any sort of questions I can answer? I know that was really quick, but I wanted to keep this under 5 minutes.
Right. I mean, again, because this is in a sense an introductory class, the focus of the class in terms of lectures is probably going to be on CompuCell. But if there were people here who were more computer engineering or computer science who were interested in core code development, or people who found this kind of modeling that Andy's talking about intriguing, you definitely have the right to work on the project with Mechanica. The thing I'd have to warn you about is that the class time will be devoted to CompuCell. And so you'd have a little bit of a disconnect because we'd be teaching you how to do certain things in CompuCell, and then you'd have to work with Andy to learn how to do them in Mechanica. And so the sort of the bandwidth would be a little higher for Mechanica project. But Mechanica is a beautiful package. Andy's done an amazing job developing it. And I definitely wanted to give people the option, if they found that intriguing, of working with Andy on that. It's also possible, of course, that that's something you do outside of the class. Again, whatever, and we're running out of time now, so we won't get to run the COVID simulation today. We'll do that at the beginning of the next class. But I hope that was perhaps a little disorganized, but I hope it gave you a set of ideas about the kinds of things you could do with this kind of modeling, and what a project might look like. And one of the things that you saw, which is why I wanted to end with the COVID simulation, was that running, you can in fact upload and run any CompuCell simulation at the moment on Nanohub. The problem is that when you do it, you have these little issues, like you have to load it with the right path and you have to run it. When you package a CompuCell simulation for Nanohub, when you click that start button, it will open up the window as you want it to be opened. And maybe we'll just end. I'm not going to be able to get everybody to do it together. But let me, and I may have jinxed myself by saying it'll work, but let's try. So let me go to Nanohub and go to dashboard. Go to dashboard for me. Oh, Nanohub. Maybe not. Why not? Stop the share. The joys of Nanohub. I think what has happened in Nanohub here, there we go, is let me just, okay, launch tool. Nanohub sometimes will only let you run, again, because it's a free application, it will only let you run have a certain number of windows open at once. So this is the COVID application. And again, it launches, when you click on it, it launches itself automatically. It knows what windows to open up and so on. It lets you change parameter values here inside of the simulation. And then it'll run. And so if you ran a CompuCell application, and packaged it on Nanohub, you could develop it either on your desktop or using that Nanohub tool that we saw. And then you would write a small script that would launch it automatically with the right configuration. And so that's something that we could then do together. We'll show, Giuliano's the expert on how to do that. And next time we'll look at a packaged Nanohub app. We'll begin that. Again, if somebody wants to work with Andy on doing this, I'd be very happy to see that. But again, I just want to say that the expectation of the course is that it's a CompuCell course. And so working with Andy is a challenge. It's a great opportunity because it's getting it at a new package, but it's a bit more of a challenge. And you're not required to do that. Just the way you're not required to work with any of the people on any of these projects. You can work on any project you want. Nevertheless, I wanted to give you a sense of that. So we're basically out of time now. I would like to open up the floor and ask people if they have any questions on any of this. It's a lot of material or comments before we break. I hope at least some of these things were intriguing to you. I hope that there was something in what we showed that was interesting to everybody. And if not, and there's a topic in tissue biology or disease biology or tissue engineering that we missed and that you're interested in, please bring it up as a possible project and we'll go forward from there. Okay, thank you again, Andy. That was great demo. Any questions or comments before we break? I know we're just out of time. Quiet. Everybody's exhausted after 3 hours. Okay, well, in that case, I will send out a little assignment. It's hard for me to send out an assignment. It'd probably be tomorrow or Thursday. It's hard for me to set up the assignments because I don't know exactly how far we're going to get. There is a homework online. The homework online, it consists of asking you to read material about this, get CompuCell running, and so on. So please do that. And I'll be in touch with you individually and as a group. If I don't see you until next week, have a good couple of days. If you want to talk to any of the people in the group or have any questions, please feel free to contact me. I always say the same thing for myself, which is I get so many emails, I get overloaded. And so if you send me an email and I don't respond within 24 hours, it's usually not that I'm trying to ignore you, but it's that I didn't see it. Because I'm so backed up with email that I don't see it. And so your course information, you have my cell phone number. And so you can always call me. Please not 3 in the morning unless it's an emergency. But I turn my cell phone off at 3 in the morning. But if you leave a message on my cell phone or text me on my cell phone, I will get back to you. So if you try to reach me by email and you don't get a response timely, you please feel free to use my phone to get to me. Okay. Well, thank you everybody for showing up. I hope most of you will show up next week again. I hope we can find some great projects and team people up to do some really exciting work. And I think it's a great opportunity to do something fun. We were very excited in the spring when this project with the students turned into this paper. Maybe we'll get some interesting results out this semester too and help people build their careers and their understanding of biology and computing. So if I don't have any more comments or questions, I want to thank you again for showing up, and I'll call an end to the session, and I'll see you next week. Thanks, everybody.